So good afternoon, everyone. My name is Andrea Bonetto. I'm the host for today's Cancer Cachexia seminar series. So today we have the distinct pleasure to introduce our next speaker, Dr. Laure Bindels. She's originally a pharmacist. I learned this recently, who graduated from the Catholique University of Louvain in Belgium in 2008. During her PhD training, she focused her research on investigating the therapeutic potential of prebiotics and probiotics in the control of tumor progression and cachexia. She then moved to the US for her postdoc training at the University of Nebraska-Lincoln, but moved back to Belgium in 2016, where she was appointed assistant professor. Throughout her career, she was awarded numerous awards, and she succeeded in securing funding from several academic and private foundations. Her scientific productivity is outstanding. She already has over 80 publications, and the number of citations is fantastic. Scopus says that she has over 5,000 citations and an H-index of 30. Dr. Bindels' research has been focused on the development of new therapeutic nutritional tools based on the gut microbiota host crosstalk in disease settings. Her main breakthroughs are the identification of a common, reproducible, anorexia-independent microbial signature in preclinical models of cachexia, as well as the demonstration that nutritional interventions that target the microbiota have the potential to decrease tumor progression, reduce cachexia, and also increase survival in cancer. So we are very happy to have Dr. Bindels with us today. She will tell us about hepatic and intestinal alterations in cancer cachexia. Please submit your questions through the chat, and at the end of the talk, I will read them out to the audience. Laure, thank you again for accepting our invitation. We look forward to listening to your talk.
Thank you very much. Thank you very much for the kind introduction. I'll share my screen. And then there we should be good to go. Can you confirm me that you see my screen?
Yes.
Perfect. Thank you very much. So thanks again to the committee for the kind invitation. It's really a pleasure to be here today. I think it's a very nice opportunity to discuss with people in the field. And so today, as Andrea mentioned it, I will discuss about hepatic and intestinal alteration in cancer cachexia. So my lab is based in Belgium. I'll try to get the pointer. There we go. So my lab is based in Brussels, Belgium, and you may know Belgium from a few famous places like the Atomium, the Mannequin Piece, or Bruges, but also some famous beverage and some famous drinks like beer, chocolate, and fries. But we are also growing some vegetables that are much less known, like Jerusalem artichoke, for instance, and chicory roots. And we, the lab I joined 13 years ago now, was very much interested in those specific nutrients because we can extract from those nutrients a fiber or some specific fibers. And a main breakthrough of our lab was the fact that you can modulate the composition of the gut microbiota using those fibers, and this can have a positive effect in case of metabolic syndrome and obesity. And so when I joined the lab, my PhD supervisor, Professor Nathalie Delzen, offered me to work in another context where we also have metabolic syndrome and inflammation, and it was cancer cachexia. And so that's really the rationale of why we start being interested in the gut microbiota in cancer cachexia. So for those of you who don't know the gut microbiota yet, so we are made in terms of number of cells, we are half microbes and half humans. Most of our microbes are present within the gastrointestinal tract, and you have a difference of density that depends from the location. When we consider how the gut microbiota can influence the host, we have several mechanisms that have been put forward. And one of the key mechanisms is the fact that microbes can have a fermentative activity. They will produce metabolites, and then those metabolites can affect host metabolism and also host immunity. So that's really a key way for the microbes to influence the host. And then there's also this concept of a gut barrier. So the gut barrier is really all the elements that are present within the intestine and that allow to keep the microbes at bay and that prevent the translocation of pro-inflammatory component from the microbes to the circulation, and that could trigger inflammation. So through the use of several mouse models, and mainly the germ-free mouse model, so those are mice are born germ-free, so without any microbes, it has been shown that many physiological processes can be affected by the gut microbiota. And so you can see here on this scheme. I have summarized different. Different processes, physiological processes that are affected by the gut microbiota, and in the intestine it's been energy extraction, but also in tissue like the liver or the adipose tissue. It has been shown that the gut microbiota can affect lipid metabolism, adipocyte differentiation, but also brain development, behavior, appetite regulation. And then there's been a few papers showing that the gut microbiota can affect the muscles. And so this brings me to introduce you to the research question we are working on, and that I will try to go through some of them during the talk today, is the gut microbiome altered in cancer cachexia? Is the gut microbiota a good therapeutic target in cancer cachexia? What about the gut barrier function in that context? Because I told you it's an important component of the crosstalk. Is there a causal role of the gut microbiota in the metabolic and inflammatory alteration found in cancer cachexia? And then finally, we know that the gut microbiota are interacting very closely with the liver. And so what is the impact on the liver? So to answer those questions, we are working mainly with mouse model. And today I will talk to you about two mouse models. So the first one is a very well-known one about C26. Which consists in the injection of colon carcinoma cells in the flank. And then we also have the BAF model, which mimic leukemia, and it consists in fact on pro-inflammatory lymphocytes, pro-Bile lymphocytes exactly, that are transfected with an oncogenic protein, which is called BCR-Bile. And so when we inject it IV, we can mimic leukemia. And so in both models, we have seen that there are some characteristics of cachexia, so meaning splenohepatomegaly, muscle atrophy, fat mass loss, and anorexia. And so in one of our key experiments, what we did is that we took the fecal content of the mice, control mice and cachectic mice in both models. We analyzed the gut microbiota composition using 16S RNA gene sequencing. It's a technique that allows you to have a global overview of the composition of the gut microbiota. And here, what you can see is a list of taxa. So those taxa in green are enriched in control mice, and those taxa in red are enriched in cachectic mice. And this is the same here in the second model we studied. And so at that time, what we did is that we were interested to know what are the common changes that we can find in both models. And we found an increase in Enterobacteriaceae. So it's a bacterial family that encompasses many bacteria that can be pathogenic, like E. coli or Shigella. And then we also found a reduction in lactobacilli, which is a family that encompasses several bacteria that has been shown to be beneficial in some specific context. And so this really drives us to test. At the beginning, we are really focusing on, can it be a therapeutic target? So meaning if we modulate the composition of the gut microbiota, can we have benefits? And so we found out that if we restore, for instance, specific lactobacilli levels, and we were able to reduce inflammation and muscle atrophy marker in the BAF model that I just presented you. And then if we modulate the composition of the gut microbiota using some specific non-digestible oligosaccharides, we found out it was also modulating some aspect of cachexia. And then other colleagues in the field have also shown that lactobacilli can inhibit cachexia using other models. So altogether, this brings me to the first set of questions. Very shortly, yes, we have enough proof, because I show you one study, but we have many more, that the gut microbiota is altered in cancer cachexia in mouse model. Is the microbiota a therapeutic target? I believe so. We have at least a few proof in animals. And so today I will discuss more into detail what do we know about the gut barrier in this context and the liver. So that's a topic we investigate around 4 years ago. We were interested to determine the status of the gut barrier in cachectic mice. And so we found out there was an increase in the gut permeability of these mice. So this was obtained during doing a functional assay. So we took cachectic mice, we gavaged them with a fluorescent probe, and we detected the apparition of the fluorescent probe in the blood. And so this reflects directly how permeable your intestine is. And you can clearly see that cachectic mice have an increase in the gut permeability. They are also characterized by a reduced cecal content, a reduced cecal tissue weight, and this was corroborated by a few qPCR markers we measure here in the ileum, and those markers refer to gut permeability, cell renewal in the intestine, and then gut immunity. And so that was done in the C26 model. And in the leukemic model, we also found the alteration in the gut permeability, in panel cell differentiation and production of antimicrobial peptides, which are important for the gut barrier, and then also a reduction in the immunity. What we found out also in this study is that if you feed mice with symbiotics, and so here symbiotics, it means one lactobacilli and one specific fiber that was selected, we could restore this gut barrier function. And in that study, we also found that when you give this mixture, you can improve muscle atrophy and you can improve survival. But in that context, we were really facing an association. So we feed some fibers, we feed some lactobacilli, we see an improvement in the gut barrier function, and we see some improvement in cachectic parameters. And so really what we were asking ourselves is that, can we just target the gut barrier function and have an improvement of the cachexia? And so that's what we try to do with those two approaches. So the first one was a pharmacological approach, and then we administer a live bacteria. So we administer twice a day peptides to cachectic mice, which is called teduglutide, and which is a GLP-2 analog, which allow to improve the gut barrier function in other pathological contexts, but we didn't see any impact on the gut permeability and on cancer cachexia. We also feed cachectic mice. We also fed cachectic mice Fecalibacterium prosnitzi. It's a bacteria that has been shown to have a beneficial impact on the gut barrier function in Crohn's disease, and we saw no impact on gut permeability and cancer cachexia. So we were, when we were testing that, as I told you, really the idea was, okay, can we block the gut barrier and then have see how it contributes to cachexia by blocking it? And when we saw that when we use those two tools, we didn't have any impact on the gut barrier, then we were thinking there's a master switch, I would say, or master regulator that we cannot, that is preventing us from improving this gut barrier function. And so at that time, we thought it could be the interleukin-6, because it's been shown that interleukin-6 drives many of the alteration that is found in the C26 model. And so what we did is that we administer an anti-IL-6 antibody to cachectic mice. So this is indicated here in blue. And we found out that this administration of the anti-IL-6 was able to counteract the increased gut permeability that was found in these mice, but it was also allowing us to reduce the level of Enterobacteriaceae that was increased in these cachectic mice. So as a reminder, when we analyzed the gut microbiota composition, we found that it was really a characteristic of cachectic mice. We also sequenced the gut microbiota of those mice, and you can see here the results in the principal coordinate analysis. So each of the dots represent one sample, and the closer two samples are from each other, the more similar is the gut microbiota composition. And then the small far away they are from each other, then the more different is the gut microbiota composition. And clearly you can see here that the control samples are located away from the C26 samples. And when we give the anti-IL-6 antibody, you can see that 6 out of 8 mice were coming back to the composition of the control mice. So clearly, this shows us that the inflammation, and that's what I put in this summary, this shows us that the inflammation is driving the gut barrier alteration and the gut barrier dysfunction. We also, I also show you with the previous slide that a nutritional modulation of the gut microbiota alteration and the gut barrier dysfunction was able to provide some benefits, sometimes in terms of muscle or fat mass loss, in some contexts in terms of morbidity and survival. So for us, it was a proof. So yes, the gut microbiota is altered. Yes, it can be a therapeutic target, but does it really contribute? Does the microbial dysbiosis contribute to the disease? And so to answer that question, we have used two protocols that are widely used in the field of the gut microbiota, which the first one we used was fecal material transfer. So in our case, it was a cecal material transfer. So you take the cecal content for control mice and from cachectic mice, and you transfer them to mice that has been treated with antibiotics. And this is a protocol we use in other other disease models, and that works well to transfer the gut microbiota composition. And so then you can attribute the effect to the gut microbiota itself. And so what we did in that case, after the transfer, is that we analyzed the gut microbiota composition. In our case, with the C26 mice, the volume of the cecal content is extremely small, and we think that is the main reason why we were not finding a convincing data in the gut microbiota analysis. So what I mean is that we analyzed the gut microbiota composition from the donor mice and from the receiver, and the transfer was not successful. We did not recapitulate the dysbiosis with such a protocol. So we decide to use another technique that's currently used in the field, is the technique of co-housing. So co-housing means that you're going to take two types of mice. So for instance, in our case, it would be control mice and C26 mice. You will house them together. And you know that because mice are coprophagic, that they will eat each other poops, and so that you can have a transfer from one phenotype to another phenotype. And so this would be like a fecal material transfer that occurs naturally. And so again, in that case, so we did that from the beginning. Mice were either co-housed or housed with mice only controlled with control and C26 with C26 one. And we analyzed the gut microbiota composition, but this co-housing was not successful to transfer the gut dysbiosis or to block the gut dysbiosis in any of these mice. We saw similar composition of the gut microbiota, either in control mice, co-housed or not, and C26 co-housed or not. So those were, of course, very disappointing results, because this is mainly the two main techniques that's been used to demonstrate the causality of the gut microbiota. So based on those results, then we have decided to change our strategy. And this started with the PhD project of Sarah Podgen. So Sarah joined the lab in 2016, and we set up the analysis, the metabolomics analysis using proton NMR to analyze the composition of cecal content in mice in blood and in the liver. And so here are the results that Sarah obtained. So here you can see on the principal component analysis on the profile of the nuclear magnetic resonance that we have a clear distinction at the cecal content level between cachectic mice and control mice. So clearly the metabolome is different. And here you can see all the results that Sarah plotted in the cecal content, in the portal vein, in the liver, and in the vena cava. You have all the metabolites that were identified, and you can see here in orange all the metabolites that are increased in cachectic mice, and in green all the metabolites that are reduced. In dark color because they're significant, the other ones are not significant. And so if you look at them, and we will focus now today on the cecal content data, you can see that we have many changes in the metabolome of these mice, and this can be summarized in a few topics, I would say, or I have split them up in a few topics. So you have a reduction in acetate and butyrate, but you also have an increase in amino acids, and then you have some change in bile acids that were detected, and I'll come back to that later. So what we did after that is that we integrate the microbiota data with the metabolomics data to try to decipher which bacteria could be responsible for some of those changes. And one of the very striking examples was here. So here you can see that acetate and butyrate are positively correlated with one specific bacteria. And so we look it up. This bacteria, it's a member of the Rhininococcaceae family. You can see that it represents 20% of the abundance in control mice, but only 5% in the C26 mice. We know from the literature that Rhininococcaceae are very high, very efficient producer of butyrate, which is one of an important short-chain fatty acids. And so we believe that in our mice, that's the main bugs responsible for the reduction in butyrate. And so as a preliminary investigation of the consequence of butyrate on the host, we have also investigated two markers on the host site that are responsible to butyrate. And it's the proglucagon and the zinc finger protein 148. And you can see that they are reduced. And so this clearly reflects the lower host impregnation in butyrate. And so we are currently investigating what is the contribution of this reduction in butyrate in this cachectic mice. The second point that was really attracting our attention is the fact that we had an increase in most amino acids. And so we have investigated several hypotheses. The most convincing one is this one, is the fact that there is an increased transit in cachectic mice. So what we speculate is that maybe we have an increase in all the amino acids because the transit time is reduced, so the transit is quicker. And so we have a passage of the amino acids quicker to the fecal content and also less bacterial fermentation. And so to test this hypothesis of increased transit, what we did is that we look at fecal content, it was reduced. We look at stomach content, it was reduced. But we also did a functional assay. So cachectic mice were gavaged with a dye, with a blue dye, and then 10 minutes after you open the mice, and then you can track how far the dye went into the gastrointestinal tract. And you can see that we have a significant and important increase in the transit of those mice. And so clearly here, our idea is that we have an increase of the transit. It's been described that this could be due to inflammation. And so we think that with this increase of transit, we can have a reduction in the bacterial fermentation of amino acids. And so to follow up on this idea, we are now focusing on tryptophan. Tryptophan can be metabolized in three main pathways, which are serotonin, kynurenine, and indole. And the indole derivatives are produced by bacteria. So what we did, what we did is that we investigate the level of these different indole in cachectic mice. And this was confirming actually our hypothesis, because when we analyzed by mass spectrometry the profile of the bile acids, of the profile of the tryptophan metabolites, we found out, like with NMR, that there is an increase in tryptophan, but also that there is a reduction in many of these bacterial metabolites of tryptophan. So this really goes in favor of this hypothesis of an increased transit, reduced bacterial amino acid metabolism. And so what has been shown is that those bacterial metabolites, they can activate a receptor that's called AHR, so aryl hydrocarbon receptor. And when we quantified those AHR agonists, we found out that they were reduced in cachectic mice. And then finally, to relate it to this topic, so CYP1A1 is a target gene that reflects the activation of AHR. And so this is really in accordance with our hypothesis. So if we found a reduction in ileum, in the cecum, and in the liver and the muscle of the expression of CYP1A1, so this clearly reflects a reduction in the AHR activity. And so this is the topic of research is led by Dr. Adeline Dolly in my team. She's now looking at what is the role of indole derivative and AHR in cancer cachexia. So as I told you, the gut microbiota and the metabolites have a close connection to the liver. And it's mainly due to the fact that the liver is directly connected to the intestine through the portal vein. And so we're really interested in order to know what is the impact on the liver. And if we look at what is already known about cancer cachexia, and this is some of the article that's been published in the field, but what we know is that in human, there's a synthesis of acute phase response protein, there's inflammation, the liver can contribute to futile cycle and to energy cost, but also from mouse study, we know that there is a mitochondrial dysfunction, hypoketonemia, and also dysfunction in triglyceride storage. We were still interested to know more about what was happening in the liver of cachectic mice. And so that was a work that was initiated by Morgane Thibault, which is another PhD student in my team. And so she performed an hepatic transcriptomics analysis in cachectic mice. And so she analyzed in total 22,389 genes, of which 1,844 were upregulated and 1,712 were downregulated. And if you look at the PCA analysis, you can clearly see that there is a major difference between control mice and C26 mice. And so we use this data set in two ways. The first way we use it was to really try to ask specific questions based on our metabolomics results. And so that's what we did here. So for every metabolic pathway that was identified with the metabolomic analysis, we were able to check our hypothesis using these RNA-seq results, but also some biochemical complementary measurements. And to make a long story short, what we found out is that there is an increase in the amino acid uptake in the liver that may lead to an accumulation of amino acids, which drive an increase in the acute phase response protein synthesis, but also a reduction in, we found a reduction in gluconeogenesis, which was going against the hypothesis of the Cori cycles. We also found reduction in glycogen and glucose that was driving an increase in exosamine pathway and a reduction in glycolysis. And we also identified two potential new pathways that could explain the lipid accumulation in these mice. So we found out there is an accumulation of O-phosphocholine. O-phosphocholine, I think. O-phosphocholine is, can be transformed into phosphatidylcholine, and we have a reduction of the key enzyme involved in the phosphatidylcholine synthesis, and this phosphatidylcholine is actually essential to form VLDL and to allow the excretion of triglycerides. We also found that there's a reduction in the carnitine biosynthesis. So this is very different from mice to human. In mice, carnitine needs to be synthesized. And so the reduction in carnitine biosynthesis in the liver is likely responsible for the decreased level in carnitine in this liver. And this could also contribute to reduce lipid accumulation by reducing beta oxidation. So that was the first way we used the dataset, the transcriptomic dataset, is that we had some hypotheses with the metabolomics, and then we were able to look if it was correct or not and support and work on our analysis. Then what Morgane did also was a more traditional approach with RNA-seq data, is that we look at which gene were enriched and especially which pathway were enriched. So here you have a volcano plot, where you can see all the genes that are downregulated in cachectic mice and the genes that are upregulated in cachectic mice. And here you have the results of a gene set enrichment analysis. So those are a summary of the pathway that are increased or reduced in cachectic mice. And as expected in the liver of cachectic mice, you have an increase of most of the pathways that are increased are related to inflammation. It was more, I would say, I think, in my opinion, it's more novel what we found in terms of downregulation. So in terms of downregulation, we saw downregulation of fatty acid metabolism, but we also found some reduction in xenobiotic metabolism and especially in bile acid metabolism. And that's actually something that intrigue us and excite us, because we know that from those RNA-seq data that there are some alteration in the bile acid metabolic pathway. We know from the metabolomic data that there are some change, but we don't know exactly which one because in NMR it's not the best tool to do that. And then we know that bile acids can be affected by the gut microbiota composition. And so that's really motivated us to look for more into what is the role of bile acids in that context. And so to present this work, I was thinking that for those of you who don't know bile acids very well, I'll do a quick introduction. So we know bile acids can control host metabolism. They can also control immunity. So bile acids are synthesized in the liver. They can be secreted in the bile canaliculus. So they are produced as primary bile acid with mainly by the expression of CYP7A1. And then once they are produced and conjugate, they can be secreted in the bile canaliculus. They are then released in the intestine, and they can be deconjugate and then they hydroxylate in secondary bile acids that will then be passively reabsorbed in the intestine. The primary bile acids can also be reabsorbed using an active transporter, and there they can activate a nuclear receptor, which is called FXR. And then this FXR can stimulate the production of FGF19 or FGF15 in mice, and that can have a negative feedback loop on the synthesis of bile acids. So it's a very tightly controlled system. In the systemic, you also have other target of the bile acids. For instance, on the muscles, you have TGF5, which is a G protein couple receptor, which can be activated by bile acids. And you also have TGF5 expressed in the bone adipose tissue, where it has been shown to contribute to bone, to thermogenesis and bone adipose tissue activation. So I think that's also very interesting connection with the Cachexia syndrome. So what Morgan Thibault did first was to check and to characterize exactly this metabolic or this alteration in the bile acid pathway. We found out an increase in the FGF15 expression in the ileum. We also found out alteration in the profile of the bile acids in the periphery with an increase mainly of the primary and the heteroconjugated bile acids, and an accumulation of bile acids in the liver, which was not due to an increase in synthesis, because synthesis was actually reduced and conjugation were also actually reduced. And so this really, so we were thinking, okay, what could explain this reduction, this accumulation of bile acids in the liver while the synthesis is reduced? And we believe the answer is an alteration in the hepatobiliary transport system. So here what you see are the results of the RNA-seq. So in the scheme, what you see is that the transporter responsible for the entry in the hepatocyte and their expression is reduced. And there are also the expression of the transporter that are responsible for the transport of bile acids and bile components in the bile canaliculus are also reduced. And then finally, we have 3 transporters which are responsible for an export from bile acids from the hepatocyte to the systemic circulation. They were upregulated as a response to this accumulation of bile acids. And so really, when we saw those results, we made a connection with other syndrome that's been described and that's also associated with inflammation. It's inflammation-induced cholestasis. So it's the fact that bile acids can accumulate in the liver due to inflammation. And one of the key clinical markers that is measured in that context is bilirubin. And so you can see here that bilirubin is drastically increased in 36 mice, which altogether led us postulate or think or demonstrate, I would say, with the accumulation of bile acid that there's cholestasis in these mice. So here it's the confirmation of those results in an independent experiment using qPCR. So we see the same alteration in the independent experiment. So that's clear. We have alteration of the bile acid pathway in cachectic mice. We also found some sign in the LLC mice, so that was quite convincing, I thought. But we were wondering what is the contribution of those bile acids to cancer cachexia. And so to answer this question, what we did is that we fed some mice with cholestyramine. So cholestyramine is a bile acid sequestrant. It's a resin that allows to reduce the absorption of bile acids, and they are excreted in the feces. And so here you can see that cholestyramine had no impact on tumor weight, but it was able to reduce FGF15 in the ileum, which is a proof that cholestyramine was preventing well the reabsorption of bile acids. We also checked the hepatic bile acid profile, and it was most of the increase in bile acids were contracted by the administration of cholestyramine. So we next wonder what is then the impact on the liver of this decrease in the absorption of bile acids. And so we used transcriptomics for that, and we found out that 48 genes were upregulated by cholestyramine and 10 genes were downregulated by cholestyramine. And among those 10 genes, we found that genes involved in hepatic inflammation were reduced, with the most important reduction observed for MMP8, for instance, and for CXCL2. The next question, of course, in mind is that what is the consequence for the host in terms of cachexia? And so here the results are, I would say, less convincing. There are some significant change in cachectic parameters, but it's not biologically that interesting, I think. So we have a reduction in we have no change in the body weight evolution in absolute amount, but when you look at the evolution over the last few days, we have a slight reduction in the body weight lost. We have no impact on food intake, but we also we have found some trends toward reduction in muscle atrophy and bone adipose tissue thermogenesis. Here, I'd like to mention that we don't know yet. So we know cholestyramine reduced the load of bile acids in the enterohepatic cycle, but we don't know yet if it's reduced the amount of bile acid in the periphery. And that's something we would like to investigate to better understand what is the contribution of bile acids to the cachexia phenotype itself. So when I say cachexia phenotype, then more traditional marker like muscle atrophy and adipose tissue activation. And then finally, we were interested to know if this alteration in the bile acid profile was also found in human, because we know that human and mice are not the same. And so we took advantage of the Actica study that was done by Professor Jean-Paul Thyssen and Dr. Audrey Louvain in our university. And we measured the total bile acid serum in those patients. So it was 94 colorectal cancer patients, 41 were non-cachectic, and 53 were cachectic, according to the Feron definition. And we found out there is an increase in the total bile acids in the serum of this patient. And we also analyzed by mass spectrometry the bile acid profile of those patients, and we found similar change that what we found in mice, meaning that we have an increase in toro bile acids or toro conjugated primary bile acids in patients. And then what we did is that we integrate this concentration of the different bile acids into an analysis that allows us to take into account age and sex, and we integrate the data with several metadata that was available for this cohort. And we found out that bile acids were strongly and positively associated with inflammatory marker and negatively associated with survival. So clearly from this work, what we know is that bile acid alteration contribute to inflammation. I didn't show you the data, but we also have proved that inflammation induces bile acid alteration. And what we are working on now is trying to understand if the microbial dysbiosis contribute to bile acid alteration, and if bile acid alteration contribute to more, I would say, more classical feature of cachexia like muscle atrophy and fat mass loss. So this brings me to the conclusion of my talk today. So is the gut microbiome altering cancer cachexia? In most models, I would say yes. Can it be a therapeutic target? I believe so. What about the gut barrier function? We don't have a clear answer yet for that. And it's more or less the same answer for the causal role for the gut microbiota. We know for sure that if we target some of the metabolites, we can have some benefits. That's what our study with the bile acids suggests. And then I also show you what we have obtained in the liver recently, and that I hope will help us to better understand what's happening in the liver of cachectic patients. And clearly, and that would be my main slide of perspective, is that what we have shown is done mainly in mice. And so we are interested to know what's happening in humans. And so that's the goal of the microML study that has been funded by the ESPEN, and that started in 2016. So this project aims to, it's a multicentric study that aims to analyze the composition and the activity of the gut microbiota in acute myeloid leukemic patient. We have collected urine, feces, and blood sample, performed nutritional assessment, cachexia symptoms. Recruitment was finished in December 2019, and then Sarah Bojan is leading this analysis, and we hope to get the data soon to share it with the community. So that said, I'd like to thank very much all the people involved in this project, all the people who decide to join my team, our collaborators, our fundings for sure, you for your attention, and I leave you with that image. I think it's interdisciplinarity that make it all the fun in science. So I'm happy to take questions if you have some. Thank you.
Thank you, Laure. Thank you so much. Really, really nice and inspiring talk. While we wait for some more questions, again, I remind you, please use the chat for the questions. I will start by asking you like a curiosity, actually. I saw that one of your last slides was presenting some the bile acid changes in across different patients with cancer. I was curious, are these changes most common in those patients that have cancers of the abdominal or intestinal tract, or they are equally occurring across different kinds of cancers? Because did you differentiate based on cancer specificity in your data?
So it's an excellent question. So here we have analyzed patients only with colorectal cancer. We haven't checked other type of cancer yet. I think it would be interesting indeed. We had to make some selection for the mass spectrometry analysis. So we focus on the biggest cohort we had in hands, because we believe that we cannot mix different cohorts. Yeah.
Okay. Let me see if there is any questions. So Frank Schaap is asking, let me see. Are the changes in bile salt levels in the circulation and liver also observed in the BAF model of cachexia?
I don't have access to the talk, and I didn't understand the question. Sorry, I don't have access.
So are the changes in bile salt levels in the circulation and also in the liver observed also in the animals, in the animal model with the BAF tumor? Can you also see those in the animals?
With the BAF tumor, we haven't analyzed them. So actually, the BAF tumor model is not a good model for bile acids because we have a huge accumulation of cancer cells in the liver. It's for the whole liver in general. We're not analyzing a lot of parameters in the liver because the weight of the liver will increase from 1 gram to 3 grams, and it's mostly leukemic cells. And so since we have this close proximity between the liver and the tumor, I don't believe it's a good model to assess if it affects bile acids. I would say so, yes, because the liver is hurt. And that's also the reason why, for instance, we didn't do metabolomics in that model, because we think it will not be relevant. It would be more like a metastasis model than reflecting alteration occurring at distance of the tumor.
Okay, perfect. I have a couple of questions from Paola Costelli. The first one is, are the alterations of bile acid metabolism occurring in the early phase of cachexia?
Thank you, Paola, for the question and for your interest. So that's something we tested. We did a kinetics experiment and we necropsied the mice at the various time points, and that is not an early phase. It's not occurring early phase. They are occurring. So here we keep the mice at D10 and they're occurring at D9, and at D8 we have the change in the profile. So Yes, we have the change in the genes involved in the hepatobiliary transport, and that also made us think that's really the key reason for the accumulation of bile acids. But in the model we have, everything is occurring at the same period, because at day 8, then they start to have the small decrease already in food intake, and then we see those change, and then they all comes together. So it's very hard to say if it's coming before. And so just a related note, we check, and it's not due to reduced food intake.
Perfect. Second question from Paula. Regarding the cancer patient study, so colorectal cancer is normally associated with cachexia when it's in the advanced stages, so when it's metastatic. Are the changes that you observe referred to cachexia or to the presence of metastasis?
For those patients, if I remember correctly, but I know we put it in supplemental, there was no metastasis in the liver. I think that was checked by the clinician, but I can double check that.
Okay. So do you know if they were treated with any chemotherapeutic? And do you know if chemotherapeutics also alter bile acid metabolism and microbiota? I would say so.
Yeah. So the first question: all the patients were recruited before any chemotherapeutic intervention, so they are naive.
So they were chemotherapy naive.
Yeah. If it does affect, if chemotherapy affects bile acids, I can't answer that. I don't know. That's an interesting point, and that's clearly something we've been discussing. Should we integrate, like you did? Should we integrate the chemotherapy in our work? But then, it makes, it makes.
I know we talked about that. too many projects,
Yes.
Question from Andy Judd. You said that you had some data to show that inflammation can drive bile acid alterations. Can you expand on that, please? What inflammation? A specific cytokine?
Yes, of course. So what we have done is that we administer to cachectic mice an antibody against IL-6, like we did for the gut barrier function. It's actually the same experiment. And then we analyzed those markers of the hepatobiliary transport system, and they were completely blocked by the administration of the anti-IL-6. What we believe we should pay attention to is that when we give the anti-IL-6 antibody by IP injection, we are blocking IL-6 mainly in the circulation. And so I think the scheme is not totally correct, I would say, because we have systemic inflammation that drives the cholestasis, and this cholestasis, by the accumulation of bile acid, foster hepatic inflammation. We don't know if it affects systemic circulation. That's something we still need to assess. We have done it because bile acids require a lot of blood. So next time we do cytokine instead.
Question from Erin Talbert. Would you advise against housing tumor mice with control mice to prevent microbiota transfer?
Could you repeat, please?
Yeah. Would you advise against housing tumor mice with control mice in order to prevent microbiota transfer?
Yes, I would. And then one additional point I would advise also, and that's our case, we house mice 2 per 2, so we cannot house them individually because it would be not ethically acceptable. Right. But we always have at least 4 cages in each group, because there's been described there are some cage effect. And so you have to be sure that what's happening is not a cage effect. And so we actually have more cases and conditions we will control for that in the analysis. But so coming back to the original question, yes, I do think we should not co-house them. And just in terms of stress also, independently of the gut microbiota, we know that the stress can be communicated. When one animal is suffering, the other is not feeling good. So
yeah, that's a good point. I have a question from Fabio Penna. He's wondering whether, considering the severe anorexia that usually occurs in the mice with C26 tumors, how much of the alterations that you observed, including the gut dysbiosis, can be accounted for anorexia instead?
It's a terrific question, because it was killing me for years before we did the experiment, at least 2 years, I would say. So we have done it for the BAF model and for the C26 model. The BAF model was only by qPCR, but for the C26 model, we did a per-feeding experiment and we check, and there's no bacteria that was changed in the C26 mice that were also changed in the per-feeding. In terms of liver alteration, and specifically for the biases, for instance, if you look at all those genes here, if you look at all those genes, so we have done it also in the per-feeding experiment, and it's only the BSEP. So BSEP is mediating the export here of bile acid. That's the only one that was for which the reduction was due to the reduction in food intake. And then the other were not due to it. And also the liver steatosis, that was something we were thinking could be driven by the anorexia, because it's been shown in malnutrition children that they can have steatosis and alteration in bile acid, but this does not appear to be the case. I think we put it in the supplemental.
Next question from Marilia. Are there microanatomical changes in liver bile canaliculi and bile duct system?
Can you repeat, sorry?
Yes, no problem. Are there microanatomical changes in liver bile canaliculi and bile duct system?
That's a really good question. We were wondering that ourselves. We haven't done, so I think a good way to answer that would be to do an immunohistology of CK19, I think. And that's a sign of bile duct, the canalicule duct reaction. We haven't done that. We have checked in the RNA-seq, and we have no sign of reaction of the canal duct. But we haven't checked by immunohistochemistry, which would be the best way, I think, to do it. So okay. We don't know for sure.
There's another question. So you showed that there are alterations in hepatic CYP enzymes. So I'm particularly interested in oxylipins, eicosanoids, etc., and their pathways. Did you see COX enzymes, eicosanoids, also CAMP, PKA pathways in your models?
That's a question we got from our reviewers.
maybe he was your reviewer.
Maybe. So I'm sorry if I don't answer it yet. I know Morgane Thibault is in the audience, so she wants to complement my answer. She's welcome to join. I think we have looked at it, and it was there are some alterations that have been reported on COX enzyme, but we didn't see them in the RNA-seq. We didn't see those changes, if I remember correctly. And so we made also the full data of the RNA-seq available to the community. So please do not hesitate to check your favorite gene. It's in table supplemental 2, if I remember correctly. You have all the 3,600 genes that provide with the fold change and everything.
Perfect. I have like a very last question for you. I saw that someone targeted FGF15 by using monoclonal antibodies, for example. Have you tried a similar approach in your model to see whether there was an improvement, at least in muscle mass or muscle function?
That was something that was really exciting me. So for the muscle atrophy is not working that way, because what has been shown is that FGF15 fosters hypertrophy of the muscles. And in our case, we have an increase of FGF15, and we have a decrease we have a muscle atrophy. So it's not going in the right direction, because there's this Benoit paper published Nature Medicine 2017. It doesn't mean that FGF15 doesn't play a role, and that's an experiment we would like to do, because we know FGF15 is not acting only on muscle atrophy. So I think that would be something terrific to test. So really good.
Awesome. Well, if there's no other questions in the chat, I would like to thank you for accepting our invitation. It was a terrific talk and a really, really nice discussion. I really enjoyed it. Your talk has been recorded and will be posted on the website. So if anyone missed it, or if you want to forward the link to anyone, please go ahead and do it. thanks again, everyone, for attending, and we will see you again next month. Thank you so much.
Thank you very much. Thank you so much.
Bye-bye.
Bye.
Stay safe.
You too.